Good morning, everyone. Thank you for joining our webinar this morning. My name is Vasundhara Behel, and I am a product manager at Gator Bio. Many of you are already familiar with Gator, but for those who are not, the following slides will give you a brief introduction. So let me briefly introduce Gator before handing it over to our guest speakers from Valitor. Gator is a part of Access Medical Systems. Our parent company is divided into two wings: Gator, the Biolayer technology company that makes products for research use only, and ET Healthcare, the wing that makes diagnostics and currently conducts business in the Chinese market. More about Gator Bio. We are based in Palo Alto, California, and our customer base is rapidly expanding not only in North America, but worldwide. We have a very strong IP portfolio with over 50 granted patents. Our R&D team and the team for biosensor manufacturing are both based at Palo Alto. We also have a team of dedicated field scientists offering support to our customers all over the US. Our BLI applications include quantitation, kinetics, and epitope binning for antibody therapeutic development. We also have products for viral vector analysis and LNP analysis in the field of gene therapy. So what is BLI, or Biolayer interferometry? BLI is a label-free technique for studying biomolecular interactions. It detects interactions by measuring the interference pattern of white light reflected from a biosensor surface coated with ligands. When target molecules bind to these ligands, they alter the optical thickness of the layer, thus changing the interference pattern. Real-time monitoring of these shifts allow precise analysis of association and dissociation rates among the ligand and the analyte molecules. Gator has leveraged the BLI technology to offer a complete solutions package, including instruments suited for different throughput, biosensors for various applications, and a software for data acquisition and analysis. So first, introducing the Gator BLI instrument. Gator offers 5 different instruments to suit different throughput needs, starting from a 4 simultaneous read instrument to a 32 simultaneous read instrument. All the instruments are fluidics free, making them low maintenance and are compatible with crude samples. This slide shows the 5 Gator instruments, with Pilot being the lowest throughput and Gator Pro being the highest. Between these, we have Gator Prime, Gator Plus, and Gator Pivot. So at this point, we have a total of 5 instruments catering to different throughput needs. Coming to our biosensors and kits, we have more than 28 at this time. These include sensors for unlabeled antibodies, for biotinylated proteins, small molecules and peptides, tagged proteins, and viral vectors. Some of our recently launched novel products include the Streptactin XT biosensor for twin strep tagged proteins and the anti-VHH biosensor. Then coming to the Gator One software, which is a tool for data acquisition and analysis. It is a very easy to use, simple software, and it is 21 CFR Part 11 compliant. It supports all our applications like quantitation, kinetics, epitope binning, and viral vector analysis. Gator also offers low binding BLI plates in 96 and 384 well formats. Tilt versions are available that allow for reduced sample volume, helping conserve precious sample. In summary, Gator enables various applications like quantitation, kinetics, and epitope binning with a variety of analytes ranging from antibodies to tagged proteins and peptides, as well as for viral vector analysis like AAV titer and percent full ratios. We support different throughput needs through a range of instruments that are low maintenance and easy to use. We also support our users with custom biosensors for their niche products. And if you're interested, you can find more information about this on our website. And you can reach out to us if you have a need for a custom biosensor that you don't see on the website currently. With this, I conclude the introduction to Gator and hand over to our speakers from Valitor.
Good morning. My name is Livia Breyer. I'm the Director of Development at Valitor, an early-stage biotech company located in Berkeley, California. Together with my colleague, Esmeralda Segura Zarate, I'm going to tell you a little bit about our innovative multivalent polymer technology that we engineered and developed with the help of our Gator BLI instrument. In addition, we are going to tell you about how we streamline the screening and characterization of a new set of VHH antibodies in an early-stage project using Gator's new anti-VHH probes. At Valitor, we have a novel multivalent polymer conjugation platform technology. The way it works is that we look at proven therapeutic targets and de-risk pathways that could be improved upon with our platform technology. We then either internally develop or use existing drug payloads, such as peptides, small molecules, nucleotides, or antibodies, to covalently conjugate to our multivalent polymer platform to create a new chemical entity that we call an MVP. This drug is tunable and engineered to be injected into the target tissue, such as an eye, for a long-acting, effective, and safe therapeutic. And at Valitor, we have an interchangeable library of polymers and payloads that can be combined to generate an entire pipeline of superior medicines. Our lead pipeline is conquering the issues of durability for wet AMD patients. AMD, or age-related macular degeneration, is a disease that affects the visions of millions of patients worldwide. There are several biologic therapeutics on the market that work quite well. However, they are either antibody or receptor decoy-based therapeutics that rapidly diffuse from the vitreous cavity of the eye when they are injected. Therefore, to be effective, patients have to return for injections every few months, which is very burdensome for them and their families. One of the main challenges to the field is to increase the intravitreal durability of these drugs so that patients can get fewer injections. We know that one of the ways to do this is to increase the macromolecular size of the therapy, because this reduces the drug's diffusion through the dense ocular tissue. The conventional way to do this is to take one antibody therapeutic and surround it with a large biopolymer. This increases the size and durability, but can often block the antibody and reduce its potency. At Valitor, we flip this around. We have one large biopolymer and decorate it with several dozen copies of the same antibody. It's very large, so it has extended tissue durability, and because of the multivalent presentation of the antibodies, it has an avidity effect that also increases the potency. Therefore, Valitor's MVP platform is an enabling technology for long-acting intravitreal biologics and could be very beneficial for wet AMD patients. This platform technology has been engineered to improve potency and durability through a few key features. First is our polymer identity. The backbone is hyaluronic acid, an endogenous biopolymer that is also a large component of the vitreous humor, where it will be injected. Second is the backbone size. The length of tissue retention is determined by the polymer size, and we can then tune the length of tissue retention by changing the polymer length. Third is our payload. For our wet AMD therapeutic, we have chosen a single-domain anti-VHH VHH antibody that we've specifically engineered to interact with and load onto our biopolymer. Last is our potency. This is determined by the average drug spacing along the polymer that we can tune up or down based on the therapeutic needs. These four features combined have given our drug, that is still in preclinical stages, a predicted 6-month readministration window, which could greatly benefit patients compared to the other drugs that are currently on the market. And it's quite apparent here that potency is a key characteristic of our drug and something that we regularly measure and monitor as we were designing this therapeutic. Next, I'll tell you how we've been measuring it using Gator's BLI technology. Reliable potency analysis is key for any drug in development, but is absolutely critical at Valitor as it has helped us better characterize and understand our drug as we've been engineering it. We've had a Gator instrument on site since 2021 at Valitor and heavily rely on it for analysis and understanding of this drug platform. Our most tried and true assay uses commercially available AVI-tagged VEGF as our ligand, which is loaded onto streptavidin sensors. We then dip the sensors into a titration of our MVP, shown here, which is not drawn to scale, for an association step, where the anti-VEGF antibodies on our MVP bind the VEGF on the sensors. Interestingly, because of the multivalent nature of the product, once loaded onto the sensors, it will not dissociate. We regularly use this method to assess lot-to-lot variability in manufacturing, as well as stability of our drug. Next, I'm going to hand over the presentation to my colleague Esmeralda, who is our in-lab BLI expert. She's going to tell you about how, with the help of Gator's new anti-VHH sensors, we worked through another potency project when we couldn't use our tried and true BLI setup.
Thanks, Livy. So on one of the projects I was working on, I wanted to test 3 different VHH antibodies up against a target protein from multiple species and from different isoforms. One barrier to this was that most of our target proteins that we wanted to test were not commercially available with AVI tags, and therefore I could not go our usual route using the streptavidin sensors. We had a few options to go about screening our VHH antibodies with commercially available ligands without AVI tags, and the first option was to biotinylate the protein ourselves. However, there were 2 main flaws with this approach. The first was, if we wanted to biotinylate the VHH antibodies, we would need to optimize tagging conditions so that the stability and biotinylation efficiency of the VHH antibodies remain the same across all VHH antibodies and lots we screen. The second was that if we wanted to biotinylate the target protein, there is a risk of biotinylating an exposed lysine residue that is key to the binding epitope. Therefore, blocking it could generate biased results. Although possible, biotinylation would be a laborious task requiring weeks of method development for tagging conditions, but that's just time we don't have. Our second option was using Gator's AR2G lysine reactive sensors. So to give a brief background on how the AR2G sensors work, pre-coded sensors are first activated to create a reactive ester. This reactive ester will then bind to lysines found on the surface of our target protein, allowing it to load onto the sensor. The quenching and blocking steps that follow take care of any nonspecific binding to any unbound esters and to the surface of the sensor. We then have our antibodies in solution for association and dissociation. This can also be done in reverse, where the antibody is bound to the sensor and the target in solution. However, this was not ideal for us, which I will further discuss later on, and therefore this was the BLI method that we used for the AR2G sensors. One drawback using the AR2G sensors is that there is the possibility of blocking the epitope of your ligand if the epitope has a lysine residue on the surface of the ligand. This was observed using a clinically available positive control that I will discuss a little later on, but we had no issues when it behaved as expected when using the AVI-tagged ligand. So luckily for us, this issue of having a blocked epitope has not been observed when screening our key VHH antibodies, so it has permitted us to use this BLI method without having to develop additional methods for tagging. One issue noticed immediately was nonspecific binding seen when VHH antibodies were bound onto the sensors and our target protein in solution. When we decided to reverse the orientation, meaning we had the target onto the sensors and our antibodies in solution, nonspecific binding was reduced, but still noticeable. We then tried using different BLI buffer conditions. Shown here are some of the alternatives we tried, including K-buffer, that's provided by Gator, a buffer with lower between 20 concentration, and another with higher salt content. None of the alternatives performed better. As seen in the graph on your right, the traditional BLI buffer we use had the best outcome, but the blank probe still had a non-specific binding signal that was roughly 30%. This was not ideal, and it took us a while to get here, but it was manageable and the best we had at the time. Another drawback using the AR2G sensors is that you can't use proteins formulated in Tris buffer if you plan to use them as your ligand. An example of that is the graph to your left. Here I ran a BLI using the amine sensors without giving a thought about the formulation of the ligand protein that I was using. So I started the BLI, I walked away for it to do its thing, and when I came back, I saw that the loading phase was extremely narrow and lasted a mere 3 seconds. Wondering why the loading phase shot up so high so quickly, I realized that the ligand that I was using was in Tris buffer. This is problematic because Tris contains primary amines that bind to the reactive esters after activation, therefore preventing ligand loading despite being diluted in BLI buffer. Having ligands in Tris would mean that we would have to do additional work to remove the Tris, such as doing a buffer exchange, but it would require more work and it would add additional variables we prefer not to have. This meant that we were not able to screen all the ligands we wanted using equivalent methods. While troubleshooting some of the issues we had with the AR2G sensors with Gator, which they have great customer service by the way, we were notified that the anti-VHH sensors have become commercially available, and so we wanted to immediately give these probes a try. So not only should the VHH antibodies and MVP platform all bind similarly, but it would require no modifications to the antibody or the ligand, so no need for ABTAC proteins. On the graph to the right is an example of a quick BLI run that we ran using our VHH antibodies as soon as we were able to get our hands on these new probes. These probes were regenerated multiple times and they produced really clean titration curves on one of our first pilot runs here. Using the VHH sensors, it allowed us to streamline the screening process, and this was because the setup was quick and simple. Compared to the AR2G sensors, I no longer had to purchase extra reagents for the activation and quenching steps to prep the AR2G probes for use. In addition, like I mentioned before, the anti-VHH probes are regenerable, which the AR2G sensors were not. The nonspecific binding is also greatly reduced when the VHH is on the sensor, which was not seen when the VHH was on the AR2G sensor, and therefore we had to work with the VHH in solution instead. There's also no need for upstream modifications, which eliminates our need for proteins to be ABI-tagged, which can be hard to find for certain proteins. There's also a reduced risk of experimental bias, as the VHH epitope is unlikely to be masked by how it binds to the anti-VHH sensors. And overall, we were able to obtain quality data within a few hours. One of the first experiments that I ran using the anti-VHH sensors was testing some of our VHH candidates up against their target protein from various species. The goal here was just to give us a quick yes or no whether a candidate binds or not. Here I graphed the response height to determine their binding to the various species of the target protein. So as you can see, VHH candidate B performed the best, not only by obtaining a response from all 4 species of the target protein tested, but it particularly produced a strong response with targets W and Y. Candidate C also performed pretty well with target W, but candidate A did not bind to any of them. Using this information and with the supporting information gathered from other internal experiments, we were able to decide to move forward with VHH candidates B and C and use animal model W for further testing. Next, we wanted to test our MVPs using the anti-VHH sensors. Our approach here this time was to run a sandwich BLI to determine if our antibody binds to the same or different epitope as commercially available antibodies. Again, we just wanted to get a yes or no answer. Do they or do they not have the same binding sites? So the design of the BLI consisted of loading the MVP to the anti-VHH sensors, followed by a target protein which would be sandwiched in the middle, then by a commercial antibody. We expected two possible outcomes using this BLI method. The first being, if our antibody and the commercial antibody had different binding sites, then it would form a complex of 3. This should then produce 3 main curves, each representing the loading of the MVP, the target protein, and the commercial antibody. If our antibody and the commercial antibody have the same binding site, then we would expect the commercial antibody to start dissociating upon loading, and it would just form a complex of 2, the MVP and the target protein only. On the BLI, it should be represented by 2 loading curves, one for the loading of the MVP and the other for the target protein, and then you'd also have a dissociation curve for the commercial antibody. Here we have the results of 2 commercial antibodies that we tested using the sandwich method. In the first commercial antibody tested, I saw the 3 main loading curves, one each for the MVP, the target, and for the commercial antibody. This shows that the commercial antibody has a different binding site to that of the MVP. The second commercial antibody tested, however, had the loading curve for the MVP, the target, and we started to get a dissociation curve for the commercial antibody. This shows that the commercial antibody was likely competing with the MVP for binding on the target protein. This approach allowed us to make and set up a complex assay simply and quickly without the need for protein modifications or nonspecific binding.
To summarize, a few key takeaways from this talk. First off, the anti-VHH sensors streamlined our BLI method development for small domain antibodies, and this is something that we had been struggling with for some time and are grateful for the innovation. In addition, this fast and straightforward analysis and experimental setup with no upstream modifications needed for our sensors made our workflow quite quick and also allowed us to decrease any bias between different antibodies or ligands in the assay. We quickly got quality data with easy regeneration, which again allowed for more replicates of this quality data. And of course, we are always appreciative of the great customer service we get through Gator. They recommended these sensors to us, helped us figure everything out, and really streamlined the process and made it easy for us to try. I'd like to acknowledge the entire Valitor team, who's pictured here, and in particular, Elena Solonenko, who's not pictured here. She ran some of the initial anti-VHH BLI experiments. In addition, I'd like to acknowledge our investors, Morningside, First Spark Ventures, the Berkeley Catalyst Fund, Pandect, and Excite Ventures. And now we can move to the Q&A session.
Thank you so much, Vasundhara and the Valitor team for a great presentation. As promised, we would like to share the exciting offer that Gator Bio is currently offering, which would be on your screens right now. So I'll let you guys have a look. So this is an exciting trade-in offer. If you're looking to upgrade your current existing BLI systems, we do have a promotion that's ongoing. So if you would like to contact us regarding the same, please scan the QR code or reach out to sales@gatorbio.com for further information. Thank you. I will quickly launch a quick poll on your screens. So if you can help answer those, and we can get into the Q&A session right after. Thank you. So the first question is for Vasundhara. Can we use anti-VHH probes on Octade BLI instruments?
No, the Gator anti-VHH probes are not compatible with the Octade BLI instrument. They are only compatible with the Gator instrument.
Quickly getting into the next one. The slide showing the curves with buffer optimization to lower the A-specific binding showed the highest increase. So I believe this is a question for your team,
Yep.
Valitor.
Yes, so those are, and those are kind of some of the standard ways that we had thought of, in communication with Gator when we were having this non-specific binding problem. But I think with a lot of this type of method development, because we didn't necessarily know what was causing the non-specific binding, some of those didn't work. And as that was clear in the method there, and that's kind of why you have to just trial and error things. But we were a little disappointed that none of our ideas improved things as we had expected. But yep, that's a astute observation.
Great. Thank you. The next question is, I believe it's for Vasundhara, but it is, what is the regeneration buffer used for the anti-VHH sensors?
Yeah, so the regeneration buffer is our standard regeneration buffer, which is a low pH buffer. So these probes can just be exposed to that low pH buffer, and that helps with regeneration. This is on our website as well. It's our standard regeneration buffer.
Yeah, and I'll echo that. That's what we use as well.
Great. The next question is there a correlation between the type of regeneration buffer used and the expected output?
I can take that. So the buffer that we recommend and that's been optimized is, as I said, the regeneration buffer that we offer. That's the low pH buffer. So that's the only buffer that is recommended for this product.
So let me see. Okay, we do have another question. How do the anti-VHH sensors work, and what was attached to the sensor and how they were developed?
Sure, I can take that. So these sensors are coated with a cocktail of monoclonal antibodies against different camelid species, against VHH from different camelid species. So even though it's a cocktail, it's made from monoclonal antibodies, and these are mixed in a carefully controlled ratio. So that's what is coated on them. That's what gives it a wide species. It helps to immobilize VHH from several species. And several lots of this cocktail were tested, and we've seen it's very consistent from lot to lot because of the carefully controlled ratios of the monoclonal antibodies.
Great, thank you. We have another question. I believe, Vasu, you can take this one. What is the species specificity of the anti-VHH?
Yeah, so it basically binds to VHH from all camelid species because of the cocktail of monoclonal antibodies.
Okay.
I'll also note that at least some of our anti-VHHs are humanized, and they're still binding. So granted, it's still camelid, but we've humanized them kind of using some algorithms. So.
Great. Okay, and we have another question. How should non-specific binding with crude media be eliminated?
So crude media, what we recommend is to pre-wet the biosensors in the blank media for about 15 minutes. That is something that will help minimize non-specific binding.
Great. We don't have any more questions at this point, but any of the participants, if you have any questions, please feel free to add in, and we can give it another 2 minutes.
I'll also note there was one that I kind of half answered in the.
Oh,
yeah. So I'll read the question.
okay.
So it was, in some experiments, the dissociation phases go up by quite a lot and do not seem to be due to electronic drift. How do you explain this? And I just asked a follow-up as what he was referring to, but I think I figured out which graphs on my own. These were the ones where we were looking at all the different approaches, I believe all the different approaches for the non-specific binding. And I don't necessarily have a great answer to that, but because it wasn't fixing our problem, we didn't dive into it too much. I know at least with a couple of those, they were slightly older buffers that we were just kind of trying to see. And so they may not have been the optimal quality. And so sometimes we do see a drift like that if the quality of the reagents you're using aren't great. So that's, in general, that's what we see, but I don't know if you have any other explanation for that on the technical team, Gator.
So you've already explained it, Livia, but one instance where that also happens is if the affinity is too tight. And then in the dissociation phase, you can also see some rebinding happening if the concentrations of the analyte are too high. So that's, in general, that's another reason why we see curves like that.
Yes, that's actually a good, because with our multivalent protein polymer conjugate, we often do see maybe a slight uptick, and a lot of it just depends on this, you have to have a really low noise signal to noise in your buffer. I know for us, we have a Gator Pro. The Gator Prime wasn't quite ready yet, but I do know the Gator Prime can really, is that true? The Gator Prime can really help with, some of that drift. At least that's what I was told.
Okay.
Great. So I think we're good. So I would really like to thank our speakers today, Vasundhara and the Valitor team, for their great presentation and answering all of the questions. And also thank you everyone for attending today's webinar. If there are any questions regarding the anti-VHH probes or the experiments, or you have questions regarding our trade and offer, please feel free to reach out at info@gatorbio.com or sales@gatorbio.com. And we would be sending a follow-up email within the next 24 to 48 hours with the recording of today's webinar. So on behalf of Gator Bio and Valitor and our presenters, thank you for joining us today, and I hope you have a great rest of your day. Thank you.
Thank you.
Thank you.